So thank you very much, and now we are moving to our next winner, the winner of the first session of Physiology, Pathophysiology and Pharmacology session, Gábor Kozák, 6-year medical student, and the title of his talk is Impact of Aging on Spreading Depolarization Induced by Focal Brain Ischemia in Rats. Please.
Thank you. Good afternoon. I would like to talk about the impact of aging on spreading depolarizations induced by focal brain ischemia in rats. Generally, spreading depolarization is a wave in gray matter of the central nervous system characterized by near-complete breakdown of membrane potential and silencing of brain electrical activity. Under normal, normoxic circumstances, in response to the increased energy demand during restoration of the ion balance, SD induces cerebral blood flow release. SDs during ischemia are associated with various types of hemodynamic responses, and the duration of SDs extends with increasing severity of ischemia, composing a continuous spectrum from transient to terminal depolarization without any reversal to the resting potential. SDs are currently believed to exacerbate ischemic brain injury via hypoperfusion. According to our current knowledge, the conversion of ischemic tissue to infarction is accelerated with age, and elderly people have to face a higher mortality and a worse functional outcome, but the aged brain has a lower susceptibility to SDs. No clinical study has systematically investigated the relationship between depolarization events, hemodynamic responses, and age. Therefore, the purpose of this study was to identify and describe emerging SDs and coupled local CBF changes following a short, transient middle cerebral artery occlusion at different ages in animal model. Male Vista rats of 3 groups of age were compared: young, middle-aged, and aged. During the surgical procedures, both common carotid arteries were separated from the surrounding tissue, and a long thick suture was placed around them in order to carry out occlusion later. A cranial window was prepared over the right parietal cortex, and it was loaded with voltage-sensitive dye. A second, smaller cranial window was drilled through the temporal bone over the distal middle cerebral artery. The experimental setup was designed regarding spectroscopic considerations. If a circulatory system is illuminated by green light, at which wavelength the absorption of the deoxygenated and oxygenated hemoglobin are the same, the intensity fluctuations of the reflected light are proportional to the changes of the local blood volume. In order to capture voltage-sensitive fluorescent images, we illuminated the cranial window with green light, with red light. And beside these excitations, we performed laser speckle contrast imaging to monitor local blood flow. The different excitations, and could you hear me with this microphone? Okay, thank you. The different excitations were applied as consecutive flashes, and images were captured with the help of 2 identical CCD cameras. After the careful preparations, 10 minutes of baseline was recorded. The focal brain ischemia was performed by placing a small microaneurysm clip around the distal middle cerebral artery, and both common carotid arteries were occluded by pulling gently on the sutures looped around them. After a 30 minutes period of ischemia, the clip was removed and the sutures were released. Images were captured for an additional hour, after which the rats were euthanized. At the end, 10 minutes of biological zero was recorded. According to previously established methods, first, the analysis of 3 randomly chosen small areas over the cranial window was performed in all sequences. First, we identified the depolarization events and characterized the waves as transients if the positive shift of the voltage-sensitive dye is followed within 200 seconds by the reversal to the baseline, otherwise they were dimmed. Accompanying CBF responses were also identified. In order to qualitatively track the spatial features of the depolarization events and the corresponding hemodynamic responses, I extended the single point analysis to the whole field of view under the cranial window using MATLAB scripts, automatically identifying and characterizing the depolarization events and the coupled CBF changes. Practically, it means the evaluation of more than 100,000 points instead of 3. On this slide, you can see representative maps from each group of age, where the green pixels indicate the areas exposed to prolonged depolarization. You can see that in the old brain, a larger area was invaded by prolonged depolarization. Regarding the frequency of the SDs, we can say that in young and middle-aged rats, there were significantly more depolarizations, and the percentage of the area of transient depolarization was significantly larger in the case of young and middle-aged animals. Accordingly, the area involved in the evolution of prolonged depolarization was significantly larger in old animals. In this slide, you can see representative map pairs from each group of age where the flow values are expressed in the percentage of the baseline CBF prior to ischemia. Values are given for 2 distinct time points: immediately after clipping and immediately preceding artery release. You can see that the extension of higher perfusion area increased during the ischemia in young and middle-aged animals and decreased in old rats. The areas with the most severe ischemia completely disappeared in young and middle-aged animals, and the share of this area extended in old animals. We saw that all of the prolonged SDs were coupled with a decrease in CBF in the case of old animals. The statistical correlation between the area of prolonged SD and the area of most severe ischemia showed a significant positive correlation, and at the same time the area of transient depolarization to the area of severe ischemia showed an inverse relationship. The severe ischemia in this case means the areas where the CBF value was lower than 20% of the baseline CBF at the end of the ischemia. As a summary, I can tell you that I developed a new analytical method for spatial and temporal monitoring of the depolarization events, and we saw that the elderly brain has fewer but often prolonged SDs and has to face a more severe ischemia. And we found a positive correlation between the area of prolonged SD and the progression of ischemia. So as a summary, we can say that SD markedly contributes to the worsening of ischemic of the aged brain. Thank you for your attention, and I would like to tell you that our laboratory is open for new students, so feel free to join us. Our department, the Department of Medical Physics and Informatics, has 2 senior mentors, Professor Ferenc Bari and Dr. Farkas Eszter, and they are very keen to teach you, and you can do cool experiments, and as you saw, you can do very important experiments. So thank you.